Hello viewers, my name is Mrs. Z Koloka from Ebenezer Majombozi High School, a life science educator. Welcome to the ECDoE broadcast studio. Today's lesson is about the paper that we are going to write next week on the 3rd of June. I will be doing an overview of the paper. The paper consists of two sections, Section A and Section B. Section A is question 1, which is out of 50 marks, and Section B is having question 2 and question 3, each 50 marks. The topic that will be examined is DNA, code of life, reproduction in invertebrates, human reproduction, genetics and inheritance, human response to the environment. And the mark allocation is as it is written on the screen. For example, DNA code of life is 20 marks, meiosis 16 marks, reproduction in invertebrates is 6 marks, human reproduction 31 marks, genetics and inheritance is 36 marks, and human response to the environment is 41 marks. And from this we can see which topic is heavily weighted and which topic we must focus on and put on more focus. Then Section A is basically multiple choice and terminology. It is important that learners, we choose the correct answer there, and choosing that correct answer, you must make sure that you choose a reasoned answer. Do not give us two answers. For example, give us A or B, because we are not going to choose for you. You must be sure of the answer that you have chosen. You must also be very mindful of terminology, as we have got some terms that are closely related. For example, the choroid and the choroid. There is a difference between the choroid, as it is the layer in the eye. Choroid, it is one of the membranes, the extra embryonic membranes in your amniotic egg. Urethra, ureter, corpus luteum, and corpus callosum. These words are closely related, but they are totally different. So let's make sure that we are very much mindful of these terms and we are aware of what they mean. In Section A also, where a question requires us to write a letter and the name, students tend to write only the name and leave out the letter. And by so doing, you are losing marks. So make sure that you follow the instructions. If the question says choose the letter and the name, if it's A, fallopian tube, according to the diagram that we are given, you say A, fallopian tube, so that you don't lose marks unnecessarily. Also, if you are given a diagram, make sure that if a question says that give the visible reasons for your answer, the examiner wants you to extract something from the diagram that we are given, not something that you know. Use the diagram that we are given and then tell us about what you are actually given on the diagram, not what you know, because we normally lose marks on something that are like that. Also, you must be able, as learners, to make sure that you note the command of words in each question. For example, there will be some questions which require you to describe a certain concept or to explain a particular concept. So therefore, you must know the difference between describe as well as explain. Because when we are required to describe, it only wants you to give details of what something is like. And yet, when you are required to explain, it requires you to tell us why that particular thing is like that. For example, a question might say, describe the role of semicircular canal in maintaining body balance when the speed and direction of the head changes. So this question wants you just to describe the role of the semicircular canals. You will begin by saying, when the speed and direction of the head changes, then the fluid in the semicircular canals will move from one plane to another. So which means you are just describing what is actually happening here, which becomes different now when the examiner says explain. The description, you are just telling us how is this process taking place. Then when they say explain, they want us to, for you to explain why is it happening like that. For example, the second question, which is related to this one, will be saying, explain how the semicircular canals are adapted for their function. The function that we have already described, which is maintaining balance. Then you begin to look at the structure of your semicircular canals. They are arranged on different planes. So therefore, as that fluid moves, it's moving from one plane to another until it reaches the ampullae where your cristae are found. Once you talk about cristae, we are also indicating that these semicircular canals at the base, they have got the receptors of balance, which we refer to them as the cristae. So now we are telling us that it is arranged on 3 different planes. At the same time, it has got the receptors of balance, which we refer to them as the cristae. So now you are able to explain how is this particular structure able to do this. The first part, you were able to describe how is it doing it. So therefore, we must be able to identify when a question says describe and explain, because we normally lose marks by not being able to answer according to what the question is asking us to do. Also, we must note that when we are looking at the topic DNA code of life, there is a difference between diploid and haploid. Remember, when we speak of diploid, we speak of a cell that has got a full set of chromosomes. But once it is haploid, then it means it has got a single set of chromosomes. So make sure that you don't mix up these terms, diploid, haploid. Diploid, both chromosomes from paternal origin and also from maternal origin are in that particular cell. It's a full set. For example, if it's a human cell, it means the cell has got 46 chromosomes. If it's haploid, then the cell will have 23 chromosomes, which means it has got a single set, not a full set of those chromosomes. Also, we must also be careful about understanding the full names of the, your nucleic acids when dealing with protein synthesis. For example, we understand that we abbreviate them by saying DNA, mRNA, tRNA. Some learners will write, instead of saying transfer RNA, they will say transport RNA. So which means somehow they are kind of like giving an incorrect name to that particular nucleic acid. Also, learners, we must be able to contextualize the aspects of mutation. This question of mutation, the learners, they always leave it unanswered, or sometimes they just generalize when they answer this particular question. It is important that if you are given a context and you are given that this mutation has occurred in this particular area, identify that particular area. If, for example, we are saying that mutation occurred on a code CAT, and the last T has been changed into A, then it means the code has changed from CAT to CAA. So you tell us where the mutation has occurred. How has this affected the codons on the mRNA? So which means if it has occurred on a DNA, the codons on the mRNA will also be affected. Also, the anticodons on the tRNA will also be affected. So which means therefore that a different amino acid will be picked up. If you are given a set of amino acids there, tell us which amino acid now has been taken instead of the original one, so that we understand fully that you understand the concept of mutation, instead of you generalizing that mutation will cause a change in the change of the codons, and therefore a different amino acid will be picked up, and yet in that particular question you are given the type of amino acid that it will change to. So please be careful about that. Contextualize your answers as you are given in the exam. Also, we look at meiosis. One of the things that we normally miss there, not because we do not have the content, but we rush when we're answering these questions. Remember, meiosis has got 2 types of meiosis, meiosis 1 as well as meiosis 2. And the phases are the same, but you need to indicate in which phase is this happening. So therefore, you must be able to indicate that is it metaphase 1, is it metaphase 2? Is it anaphase 1? Is it anaphase 2? In most cases, you'll be given diagrams. For example, in the diagram that we have here in front, there are some chromatids that are pulled to the opposite poles. So which indicates that this is meiosis 2. And which phase of meiosis 2 is this one? It is anaphase 2. Why are we saying that is anaphase 2? If you look at it, chromatids are being pulled to the opposite poles. And yet during meiosis 1, a full chromosome is pulled to the opposite pole. So we must be able to say that it is meiosis 1 or it is meiosis 2. Make sure that you do that. If you are required to draw a phase after this one, don't come up with your own chromosomes. Please use the chromosomes that you are given. If the chromosomes are shaded like this one, make sure that the chromosomes that you have in your own drawing are also shaded the same way, because you lose marks. It will mean you're bringing up now chromosomes with a different genetic material, and yet you have been given a particular template in your exam. So please, learners, let's make sure that we are not losing marks on things that we can easily get marks on. Then also, once a question asks you to describe the role of structure B in cell division, then you look at structure B. What is structure B? Your structure B there, it is your spindle fibers. What is its role? Remember, it works during anaphase 1 or anaphase 2, where it contracts and then pull your chromosomes to the opposite poles. It's either pulling the chromatids to the opposite poles if it's meiosis 2, but it's pulling chromosomes to the opposite poles if it's meiosis 1. So please be very mindful about that. Then also in human reproduction, learners, there's another thing that we need to note here. The structure of an ovary is totally different with the structure of a Grapharian follicle. You normally confuse this structure that is here with the structure of a Grapharian follicle. And it is important that whilst we are learning here, we must also make drawings for ourselves. When we explain the concept of ovulation, you can see that B, the structure B there, it is your ovary. And then we can see that here is an ovum moving from structure B to structure C. So the movement of that ovum from your ovaries to your fallopian tube, it is the process of what? Of ovulation. So it is important that we don't confuse the process of ovulation or the structure of your ovary with the structure of your Grapharian follicles. It is important that we use diagrams whenever we are dealing with this area so that we are able to understand the differences between these diagrams. Also, another headache for us as matriculants, we always have a problem in understanding the role of hormones in menstrual cycle. One of the things I normally advise learners to do is for us to understand what is menstrual cycle. So the simplest way I normally say the learners should explain it, it is by saying that it is the changes that occur in the ovary as well as in the uterus over a period of 28 days. That's how you describe it. And then you begin to say which hormones then are involved there, or what changes are occurring in the ovary. The minute you talk about the changes that occur in the ovary, it means now you are describing part of menstrual cycle, which is an ovarian cycle. You begin to tell us what is actually happening during ovarian cycle. One of the things that's happened there, it is the development of follicles. Once the follicles are fully developed, the next thing that is going to happen, the matured follicle will rupture and release an ovum. And then that process, when a matured follicle ruptures and releases an ovum from the ovaries to the fallopian tubes, that process is known as ovulation. And it is occurring in the ovaries. Hence, we are saying that it is a change that is occurring in the ovaries. Also, once those Grapharian follicles are ruptured, then they change form now. They will change their form from being Grapharian follicles to a corpus luteum. So therefore, learners, these are the changes that actually happen in the ovaries. These changes are not occurring on their own. There are hormones that are influencing them. Once you speak of the development of follicles, you know that the hormone that is responsible for the development of follicles, it is your FSH. So which means therefore that the FSH, follicle developing hormone, is responsible for developing what? Your follicles. Then once the follicles are fully developed, another hormone now will take in, which will cause that matured follicle to rupture and release that ovum. And that process we refer to it as ovulation. And the hormone is called luteinizing hormone. In short, it is called LH. Both of these hormones are secreted from the pituitary gland. It is important to know the source of the hormone and where the hormone works. It is working in the ovaries. What is it doing? It is developing the follicles. That is FSH. What is LH doing? It is influencing or stimulating the process of ovulation. Once the process of ovulation is done, then that Grapharian follicle is converted into a corpus luteum. LH is responsible for that. So there are two functions of LH, that of facilitating or stimulating ovulation and also of stimulating the conversion of Grapharian follicle into a corpus luteum. Then we are done with the first part of your menstrual cycle. Then the second part, it is the uterine cycle. Then I normally say to students, one of the things that you should notice, the uterine cycle are the changes that occur in the uterus. What are these changes that occur in the uterus? One of the things that happens there, remember the uterus is lined up with a membrane that we refer to it as the endometrium. So that endometrium that lines up your uterus thickens. And as it thickens, it becomes glandular. And as it becomes glandular, then it also becomes vascular. Why does it become glandular? Why does it become vascular? In preparation for pregnancy. So which means therefore that once an egg is released from your ovaries, your body is thinking that you are going to be pregnant. So therefore it must prepare the uterus. How is it preparing it? By thickening it, by making it glandular, by making it vascular. Who is responsible for that? The first hormone that is responsible for that, it is the hormone that we refer to it as the estrogen. Where is the estrogen released? It is released from those Grapharian follicles. So this type of hormone is released by the Grapharian follicles. Then the second thing that happens is that after that, then the endometrium yako will become more thicker, more glandular, more vascular. What causes it to be more glandular, more thicker, more vascular? It is the progesterone. Where is the progesterone secreted? The progesterone is secreted from the corpus luteum. Remember, the corpus luteum was formed from the remains of the Grapharian follicle. Then it takes over the function of the estrogen and therefore continues to make sure that pregnancy is maintained. Let's say, for example, you are already pregnant, so you must not lose that baby. So that baby must remain in your uterus until its term is over. That is, the gestation period is over. So for it to remain there, the endometrium needs to be more glandular, more vascular, more thicker. What makes it like that? It is the hormone that we refer to it as progesterone. What is the source of progesterone? It is the corpus luteum. Another source of progesterone, it is the placenta. Remember, when that developing embryo is attached into your endometrium, it is attached by the placenta. So therefore, the placenta will release progesterone in order to maintain pregnancy. Another thing, as we learn about these hormones, we need to know the negative feedback between the progesterone levels as well as the FSH levels. For example, a question might come in an exam and ask you, why are women given pills with progesterone in order to prevent pregnancy? And then you begin to say that they are given progesterone, a pill with progesterone level, because high amounts of progesterone level inhibit the pituitary gland from secreting FSH, because the person does not want to be pregnant. So if the person doesn't want to be pregnant, then the FSH must not be secreted, because if the FSH is secreted, it is responsible for the development of follicles. So if the follicles develop, then it means therefore that the ovum will be produced. So to avoid the ovum or the eggs to be produced, then the FSH must not be secreted at all. So high amounts of progesterone will inhibit the pituitary gland from secreting the what? The FSH. So that is the functional relationship between the progesterone and FSH. If the levels of progesterone are high, then the pituitary gland will be inhibited to secrete what? FSH. And as on genetics and inheritance, one of the things that we have noted that you sometimes fail to differentiate between the genotype as well as the genotype of gametes. That is genotype of organisms or offsprings and the genotype of e-gametes. For example, in the example that I have in front of me here, it says in certain species of butterfly, one gene controls wing spot color and another one controls eye color. The wing spot color can be red spot or gray spot. Already we are given the alleles there, capital letter R, and the other one we are given the allele small letter r. So which means therefore that those are the alleles or the genes that you are going to use for the genotype of that particular offsprings. While the eye color black is represented by an allele E and brown is represented by an allele small letter E. Once you see a capital letter, it means it is referring to a dominant allele. A dominant allele is the allele that masks the recessive allele in the phenotype, especially if it's in heterozygous form. So that one will be dominant. And then a recessive one is the one that is masked, and we always represent it with small letters. If we look at the table that we have here, it says, the table below shows the phenotypes and all the possible genotypes of offsprings. We have got red spots and brown spots, and then there is a missing information there, which is X. Then let's try and fit in all the possible genotypes of OX. Red spots, we are already given that red spots is a dominant trait, so which means therefore that for red spots, we'll have capital letter R, capital letter R. Or if it is heterozygous, we might have capital letter R and small letter r for the red spots. Then for the brown spots, we have already been told that brown spots is a recessive allele, and therefore we'll represent it by small letters. And therefore it will be small letter E and small letter E. Those are the ones that you can put in to get that particular mark. Then the phenotype of Y. The genotype is given there is capital letter R, small letter r. Capital letter E, small letter e. So which means therefore that now, if it's capital letter, it means it's representing the dominant trait. What is the dominant trait according to this example here? It is the red spot. So therefore, this particular organism that is under question here has got red spot. And then we look at the second part, phenotype of the second trait that it has there. It is represented by the genotype capital letter E and small letter e. And then we look at the what we are given there. It says black is represented by E. So which means therefore that if it has got a capital letter E, black is dominant over brown. So this particular one has got red spots with black color of the ears. Okay, learners, we have finished the first part of this lesson. We will still be continuing with this lesson on genetics on the next lesson. Thank you, viewers. We have come to the end of our lesson. You can leave your questions and comments. Don't forget to leave your name of the school so that you can give shout-outs. Please share and recommend and subscribe to our YouTube channel. Thank you for watching. See you in the next lesson. We will still be continuing on our overview of paper 1.